So now that we know how to calculate allele frequencies and genotype frequencies, we're ready to discuss the test for whether a population is evolving or not that I referred to back from part one. It is now part two of the chapter 23, Evolution of Populations. As I have said previously, just because genetic variation is required for evolution to occur, it does not mean that having genetic variation always result in evolution. We actually have to test that. So if a population is not evolving, the allele and genotypic frequencies should remain constant from generation to generation. Such a population is what we call in Hardy-Weinberg equilibrium. We'll talk about what this means in a bit when we explain what is the Hardy-Weinberg equation. So to determine whether a population is at what we call Hardy-Weinberg equilibrium, given that we just have two alleles at a given locus, we can apply the concept of calculating genotype and allele frequency to the Hardy-Weinberg equation. So again, this is only usable when we're talking about just two alleles at a given locus. We either have big A or little a, or maybe CR and CW that we talked about from the wildflower population. We can't use genes that have 3 or 4 or 5 multiple alleles, like those height ranges we talked about earlier, or ABO blood type. Those are more than 2 alleles, so we can't use the Hardy-Weinberg equation. Okay, it is only for those with 2 alleles at a given locus. So in the Hardy-Weinberg equation, we're saying that it is p squared plus 2pq plus q squared equals 1. So what does this mean? Kind of like, back in the day where you have to, or like when you learn math, you have, when it got to, quadratic equations, you have to remember that formula for that particular equation. Hardy-Weinberg, again, is an equation that you have to remember as well. So make sure you remember this for the exam purposes and how to use it to calculate whether a population is evolving, or there's a second purpose as well to predict what percent of the population should have this particular genotype. We'll talk about those examples in a bit. So what does all this P and Q mean? So P is equal to one allele, frequency of the one allele, which is, if we put it into the wildflower population's case, P would be the CR allele, while Q is the frequency of the other allele. Remember, we're talking about 2 alleles at a given locus. So going back to the wildflower plants as an example, we have CW allele. So P squared then is equal to one of the homozygous genotype, which in the wildflower's case, it will be C big R, or CR. 2pq is equivalent to the frequency of the heterozygous genotype. In the wildflower's case, this will be CW, and q squared is then equal to the frequency of the other homozygous genotype. The other homozygous genotype in the wildflower's case, this will be CW. That's what all this P and Q means. So again, P represents the CR allele for the wild popular population's case. Q represents the CW allele. So if you're CR, then that is P squared, okay? If you are CW, that is 2PQ, okay, which is the P and which is CR and CW. If you're Q squared, then that is the CW genotype. And all of those are supposed to equal to 1 if the population was not evolving, okay? Not evolving. That's what we call Hardy-Weinberg equilibrium. Again, we'll apply it to a specific example in a bit. So with our calculated allele frequency for our wildflower example that we talked about earlier, we said P would then, we said CR allele frequency was 0.8, CW allele frequency was 0.2. So P would then be equal to 0.8, Q would be then equal to 0.2. Now we apply them to the Hardy-Weinberg equation to see what proportion of the population should each of these 3 genotypes would be if the population is not evolving. Again, we're using the Hardy-Weinberg equation to predict the proportion of the population with these the three different phenotypes, okay, or the three different genotypes if the population is not evolving. Remember, the Hardy-Weinberg equation is p squared plus 2pq plus q squared should equal to 1. I don't know what happened to the pluses here. Equal to 1. So if the population was not evolving, it would equal to 1. With p squared being the expected frequency of genotype CR for a wild popular population. Okay, again, this is just an example because p can be for, let's say, bushy eyebrows, and Q could be for thin eyebrows. And those, it can be for a different problem in this case. But we're talking about for wildflower population for the ease of transitioning all those calculations we did earlier with the genotype frequency and allele frequency. So expected frequency of CR would then be your P squared. And then expected frequency of genotype CW would then be your 2PQ. Expected frequency of genotype CW would then be equivalent to Q squared. So hence, if P squared is what we call, back from the wildflower example, remember P is equal to 0.8, as we've talked about, mentioned here. So P squared is equal to 0.8 times 0.8, which is equivalent to 0.64. P squared means P times P, okay, in case those of you don't know. So when we convert 0.64, we would get that 64% of plants in the next generation should have CR genotype. If we take 2PQ, okay, which is 2 times P times Q, and we already said Q is equal to 0.2. So that is 2 times 0.8 times 0.2. So that would be 0.32 or 32% of the wildflower plants in the next generation will have CRCW. Q squared, lastly, will be equivalent to just Q times 2, Q times Q, which is 0.2 times 0.2, which you will get 0.04 or 4% of the wildflower plants in the next generation will have CW if the generation has not evolved, and that's the prediction for how much of the next generation would have those three different genotypes according to the Hardy-Weinberg equation. So hence, P squared is equal to, for any given allele frequency where there are only two alleles at a given locus, we can actually use the Hardy-Weinberg equation to predict proportion of the populations in the next generation with each of these three genotypes if the species is not evolving, okay? That is the key phrase here, okay? Because if the original question is this population evolving, then that we can't use this equation just yet to predict. We have to make sure that the population is not evolving first, and then we can confirm and predict the next generation's percentage of these three different genotypes. So the Hardy-Weinberg equation allow us to calculate the expected genotypic makeup of a population that is not evolving at a particular locus. Okay, again, I repeat that. Hardy-Weinberg equation allow us to calculate the expected genetic makeup for a population that is not evolving at a particular locus. So if, let's say, the observed genetic makeup of the population differs from the expected values calculated with the Hardy-Weinberg equilibrium or equation, then the population may be evolving. Okay, so we're talking about, let's say you have let's say that wildflower population we're talking about earlier in this particular slide, and we said 64% should be red, okay, red flower. But let's say the next generation turns out to be not the case. Somehow the observed genetic makeup of the next generation of wildflowers produce 80% red flowers. Okay, so that is what differs from the expected values. So we will say that, okay, then the population is evolving. Now, but if the observed genetic makeup of the population is the same as the expected values calculated with the Hardy-Weinberg equation, then the population is not evolving. Okay, so let's say again with the wildflower population example, we said 64% of the next generation should be red flowers. So let's say we did go through the next generation of wildflowers and we counted how many of the wildflower population was red color, and it turns out that it is, let's say, 64.5%. Okay, or 645 red flowers are about 1,000, for example. Then we will say that it is what was expected from the calculation. So we will say that it is not, the population is not evolving. So make sure you remember this part for whether the test, whether a population is evolving or not, depending on what is observed versus what is expected according to the Hardy-Weinberg equation. So now let's try to apply the Hardy-Weinberg equation to another problem, because that was just the wildflower population we're talking about. So now we're going to switch over, just so you're not just thinking about a wildflower population. Like I said, the P and the Q can be any two alleles within a given locus of any population. Okay, so here's a case scenario. A locus that affects susceptibility to degenerative brain disease has two alleles, big B being the normal allele and little b being the disease allele. So let's say in a population, we have 16 individuals with genotype big B, and 92 individuals with genotype big B, little b, and 12 individuals with genotype little b. Now the question is this population evolving? Okay, so we have, again, a different scenario here, and we have two alleles, one normal, one disease for degenerative brain disease. We want to use the Hardy-Weinberg equation to see whether this population is evolving. So first, let's calculate the allele frequency based on this data that is given here. So we said we have 16 individuals with big B, 92 with big B, little b, and 12 with little b. So because we have to know the total amount of individuals first, that's why we add them together we have a total of 120 individuals in this given population. So each individual, remember, has two alleles in a genotype. So that's why we take 120 multiplied by 2 to get 240 total alleles exist in this population. For B allele frequency, we have 32. That is the big B from homozygous dominance. So 16 times 2 gives us 32. And then we have 92 individuals who were heterozygous, but still have that big B allele. So 92 from the big B allele from the heterozygous. So 32 plus 92 is equal to 124. Remember, we have a total of 240 alleles, so 124 of those are big B allele frequency. So that's what we end up with. If you take 124 divided by 240, you get about 0.52 as the allele frequency of big B, or 52%. Whereas little b allele frequency would then be, again, the 92 of them from the heterozygous individuals. 24 of them have homozygous recessive with the little b. So that means they have two B alleles each. 12 individuals times 2 gives you 24. Okay, so 92 from the heterozygous, 24 from the homozygous recessive gives you about 116 in alleles that are little b compared to the total 240. So 116 divided by 240 is equal to 0.48. That is allele frequency for little b. So based on the Hardy-Weinberg equation, if the population is not evolving, the homozygous big B genotype frequency should be P squared. So we already said this is your P, this is your Q in that sense, okay? So P squared is then taking that 0.52, this 0.52 times by 0.52 is what P squared is equivalent to 0.27. Big B, little b genotypic frequency, genotype frequency, or heterozygous is using 2PQ, and we already said Q is equal to 0.48. So 2 times P, which is 0.52, times Q, which is 0.48, is equal to 0.50 as their heterozygous genotypic frequency. And the last one is then little b genotype frequency is taking Q squared, is Q squared. Q is equal to 0.48, so 0.48 times 0.48, Q squared is then equal to 0.23. Okay, so P squared is equal to 0.27, 2PQ is equal to 0.50, and Q squared is 0.23, which essentially means 27% of the population should have homozygous big B genotype. 50% of the population should have big B, little b, heterozygous genotype. 23% of the population should have little b, homozygous little b genotype. So now we have genotype frequency of the population if the population was not evolving. We can now calculate the number of expected individuals with each genotype. Remember, total number of individuals in this particular population was 120, as we have talked about earlier, okay? So we take 120 of that population, multiply by 0.27, which is a 27%, should be big B genotype, which should be 32 individuals. And then 120 times 0.5 or 50% with heterozygous big B, little b genotype, that is equivalent to about 60. And then lastly, you have those that individuals should have homozygous little b genotype, little b, and 120 multiplied by 0.23 gives you 28 individuals with little b genotype. So if we compare these are the expected numbers from according to the Hardy-Weinberg equation that we just used to calculate, and we compare these with the observed number of individuals given in the problem, which is 16 big B, 92 big B, little b, 12 little b, obviously it is different from the expected values. We said 32 should be big B, but the population of 16. We said 60 should be big B, little b, but there's 92. We said that 28 should be little b, but we have 12 according to what's given in the problem here. So as a result, we say that this population is not in Hardy-Weinberg equilibrium and maybe then hence evolving at this particular locus for degenerative brain disease. Okay, so that is an example of how we can use the Hardy-Weinberg equation to see, first of all, predict what amount of individuals within a population should have any of the three genotype here, and then compare it with the actual to say whether the population is evolving or not. All right, so it turns out that Hardy-Weinberg equation can serve two purposes, like I was saying earlier, depending how you use it. So other than testing whether a population is evolving or not, the Hardy-Weinberg equation can also be used to predict the proportion of a population who are carriers of a disease in medical applications, because that will be useful to know if you want to know the statistics of how likely this population should have this particular disease, okay? So let's use that purpose of Hardy-Weinberg equation with an example. So in this particular case scenario, we are exploring the condition called phenylketonuria, or PKU as an abbreviation. This is a metabolic disorder that results from a recessive allele. So again, this is recessive allele condition, which means genotype for individuals with PKU is homozygous recessive. If left untreated, PKU can result in mental disability and also other problems because it tends to accumulate in the brain for people that can't digest or use break apart phenylketonuria. In order for use amino acids, sometimes we have to break it apart. So in this particular condition, so these individuals cannot use this particular amino acids. So, and it actually gives you a consequence that is not reversible. Once you have that mental disability, which is destroy or sort of impair neurons, neurons are a part of the body that cannot be regenerated once is damaged. So that's why, you want to detect this disease early so you can prevent the worst consequences of this particular condition. So this disorder occurs in 1 in every 10,000 babies born in the US. So that is your one of the given statistics right here. So now they ask you to predict the percent of population who will be carriers. So this is just talking about those with the disorder, 1 in 10,000. Now it's asking, well, what percentage of the population will then just be carriers, don't express the disease, but are heterozygous for the particular disease. Remember, carriers always means heterozygous, okay? Don't forget this definition, all right? So it says in the case scenario that disorder occurs 1 in every 10,000 babies. And disorder only occurs when one has a homozygous recessive genotype. So let's use Q, okay? So P usually is the dominant, Q is usually for the recessive. So if we're talking of the homozygous recessive genotype, we're talking about Q squared is the value. So Q squared is then equal to the amount of individuals with the disorder according to the Hardy-Weinberg equation that we just talked about. Remember, P is the allele frequency for one of the dominant alleles, and Q is the recessive, and then 2PQ is the heterozygous. So we know that if we take 1 in every 10,000 babies, that is your Q square value. If you use a calculator to divide that, you get 0.001. In order to get Q, because this is a square, in order to get Q, you would take a square root of 0.001 and turns out to be 0.01. In a sense, this is then equals to the disease allele frequency. And so we know that P plus Q is equal to 1. We talked about that before. So then if we were to solve for P is equivalent to 1 minus Q or 1 minus 0.01, which is 0.99. That is then the normal allele frequency. So by the Hardy-Weinberg equation, 2PQ is the frequency of heterozygous genotype, or what we call carriers of the disease. So now all we have to do to know what percent of the population would be carriers is take 2 times P times Q. So 2 times P, which is 0.99, as we have talked about here, and then times by Q, which is what we talked about here with 0.01. So 0.99 times 0.01 is equal to 0.02. 2 times 0.99 times 0.01 is equal to 0.02. Turn that into a percentage, then that will be 2% of the population will be carriers, okay? So you see how we can use the Hardy-Weinberg equation to predict what percent of the population will be a specific genotype. In this case, we're talking about those that are heterozygous or carriers. This is, again, assuming that the population is not evolving. Remember, we can only use the Hardy-Weinberg equation to predict the percentage of a population with particular genotype only if the population is not evolving. We'll talk about these conditions again in a bit. What conditions do we have to satisfy in order to use Hardy-Weinberg equation? And here they are. So remember we said Hardy-Weinberg equilibrium describes a population that is not evolving. So there are 5 total conditions that the case scenario must satisfy. So first of those is that there can be no mutations. Okay, why? The gene pool is modified if mutations occur or if the entire genes are deleted or duplicated. Now we can be talking about more than 2 alleles occurring at the same time, which then dissatisfy the Hardy-Weinberg equation criteria, where only 2 alleles should be participating in this whole scenario. Second, random mating must happen. If individuals mate with a subset of population, talking about that they only mate with near neighbors or close relatives, such as inbreeding, then random mixing of gametes does not occur, okay? And genotypic frequencies will change. You can't have non-random mating, okay? You can't have one population preferring to mate with certain other subset of the population, like let's say redbirds only prefer to mate with redbirds. Then that is not that is not satisfy the Hardy-Weinberg equilibrium. Okay, so it has to be randomly mating. Okay, random mating must occur. Third, no natural selection should be happening. Allele frequency change when individuals with different genotypes show consistent differences in their survival or reproductive success. Because that's the whole point of natural selection, is changing the allele frequency, right? So if natural selection changes the allele frequency, then you cannot use the Hardy-Weinberg equilibrium, okay? So no natural selection should be happening in order for us to use the equation. Fourth, extremely large population in size is needed for us to use the Hardy-Weinberg equation. Why? Well, because in small population, allele frequency can fluctuate by chance over time, and we call that genetic drift. We'll talk about specifically what genetic drift is in a little bit, in the later part of the chapter. But you have to have a large population when we want to use Hardy-Weinberg equation. You can't just say, oh, there are 5 individuals in this particular population, and I'm going to use the Hardy-Weinberg equation to calculate prediction of the 3 genotypes. You can't, because 5 is too small. You need a large population in size, like hundreds, okay? So that's another criteria for Hardy-Weinberg equation. Lastly, the 5th one is that no gene flow can be occurring. We'll talk again, just like genetic drift, we'll talk about specifically what gene flow means. For now, we'll just say that if we move alleles into or out of the population, gene flow can alter allele frequency. Again, we'll talk more about what gene flow is later in the chapter, and you'll understand why all these this will dissatisfy your Hardy-Weinberg equation if we have gene flow occurring. Okay. So remember, these are the 5 conditions that must be satisfied in order for you to apply the Hardy-Weinberg equation to a population for calculating prediction of the population having any other genotype, etc. Okay. All right. So in nature, allele and genotype frequency of a population often do change over time. So such changes can occur when at least one of the 5 conditions is not met. So if any one of these 5 conditions here that we just went through is not met, okay, this condition is not met, then we cannot use the Hardy-Weinberg equilibrium to predict next generation of offspring. Okay. All right. So if we revisit the PKU disease calculation, and we said that 2% of the population will be carriers, this 2% only is valid if the whole situation about PKU satisfies the 5 conditions of Hardy-Weinberg equilibrium. So we first of all, we said that there should be no mutations. So if we were to say that this 2% calculation is valid, no new PKU mutation should be introduced into the population, or else this 2% calculation is not valid. Second, random mating must occur, saying that people should not choose or will not choose their mates based on whether they have PKU or not. Because if people tend to choose those that don't have PKU, then that dissatisfies the random mating condition of Hardy-Weinberg equilibrium. Or if they say they will not mate with close relatives, then that's also a dissatisfaction of this condition. Mating must be random for Hardy-Weinberg equilibrium to be applied. Third, no natural selection. So we have to ignore any effects of differential survival or reproductive success among individuals with PKU. Because if, let's say, PKU gives the individual more reproductive success or less chance of survival, then that's natural selection. It will select out PKU if it's less chance of survival. So we can't use it if it has natural selection occurring. Fourth, remember that we have to have extremely large population size. We can't have anyone from this PKU gen population, right here, this PKU disease population, moving to a new small isolated location. Because if they're moving to a new small isolated location, let's say 2 individuals move from this population to the next, then we cannot trust that 2% population answer that we had earlier. Lastly, we said no gene flow. So no other population is moving into the US in this particular PKU disease scenario, okay? PKU disease scenario, okay? If you have introduction of other people moving in to this particular population, then that is gene flow, introducing new alleles in the population, okay? So nobody can be moving if we're talking about no gene flow. So now that we understand the Hardy-Weinberg equilibrium and describes a population that is not evolving, in order for a population to evolve, then it must have a change in its allele frequencies. We already said that. So how do allele frequencies within a population change, thereby resulting in evolution? Well, there are 3 possible mechanisms: natural selection, genetic drift, and gene flow. So these are all the 3 we talked about that would dissatisfy the Hardy-Weinberg equilibrium, because then if they have any of these 3 things, allele frequency would change, okay, allele frequency would change, which means then the population could evolve if the allele frequency changes. So in this diagram, it summarizes what these 3 mechanisms do to this population of beetles. So this is, let's say, the original population of beetles, where it has all different shades of color or color brown within the whole population. So first, if we're talking about the first reason, that mechanism that can alter the allele frequency, let's say natural selection. This is a concept we've talked about already extensively in the previous chapter. So let's say, for example, the more visible darker beetles that we see in this particular population is easier for predators to spot, and we're mostly the ones that are getting eaten. So this caused then the lighter beetles to have more survival or higher chance of survival over the darker beetles. And those alleles then will carry and give them that lighter coloration will become more common over time, resulting now you have a population of beetles that are mostly lighter color and not much of those that are darker color from the original population. So that's how natural selection can change the allele frequency of this particular beetle population. Second, you have genetic drift. So this particular population of beetles can evolve by genetic drift, and we will dive deeper into this new concept in the next few slides as I discuss each of these mechanisms in depth. But for now, we will say that genetic drift means that by chance alone, the alleles that give beetles darker color, let's say, have become more common over several generations. So that's why here you see more darker brown beetles in this particular population. The key word to genetic drift is by chance. There is no way of predicting which color will be more prominent in the population. because it is by chance. I'll talk more about that later. So third is that this population of beetles can also evolve by what's called gene flow, which means that let's say there's a different spotted color right here of different spotted color pattern that have arrived to this beetle original population. So in a sense, these beetles have brought over or introduced new alleles into this original population. So that's why you end up now, not just that you have dark beetles, lighter beetles, now you have spotted beetles within the population as well. That's what gene flow means, that alleles are being introduced into the population, thereby changing the allele frequencies. So now let's look at these three different mechanisms more in depth. Going in the same order as we have introduced these mechanisms that can alter allele frequency, we will start with natural selection. So remember, natural selection is based on differential success in both survival and reproduction. So this means that individuals with that exhibit variation in heritable traits, they have to be heritable. Okay, we already said non-heritable traits can't be counted into evolution. Next, traits that are better suited for the environment will produce more offspring. That's the whole point. Not only do you survive better, but you also end up reproducing more. Third, selection results in alleles passed to the next generation in proportions different from the current generation. Let's say because one allele is preferred or more favored in this environment over the other, you will not have the same allele frequency in the next generation. Okay? All right, so for example, fruit flies have an allele that is resistant to insecticide DDT. Prior to DDT use, this allele frequency was actually 0%. Okay, but after 20 plus years of DDT use, this allele frequency has increased to 37%. So in a sense, DDT was a strong selective that resulted in this frequency change. Or you can say that the environment has now changed. Before, the environment for fruit flies have no DDT. Now you change the environment to have DDT. So DDT became that selecting matter factor that will only those that have resistance to DDT will survive. Those that don't have this resistance will die off. So this is illustrated in a similar manner in this particular diagram right here, picture. So this right here is the first generation of bugs, with red ones being resistant to DDT and white ones, let's say, represents a non-resistant. So when they are first sprayed with DDT, as you can see with this little spray bottle right here, a lot of the white bug have been killed off by DDT. So leaving that only one red bug that is resistant that was in the original population to survive, and a lot less white bugs is surviving in this population. So in the second generation of this particular bug, now you already have less white bugs compared to red bugs, because from this parental population, now you have the second generation. Now again, you have you spray this second generation of bugs with DDT. Now this second generation will have left with 4 red bugs, because those 4 will not die. They are resistant to DDT. So those will remain, and then only 1 white bug may remain. I mean, they're not 100% effective. So let's say 1 does remain. So upon exposure to DDT, it changes allele frequency of red bug from small numbers to large numbers in the population. This whole population has now shifted to have more red alleles in the population, which is that DDT resistance. Thus, natural selection can cause what we call adaptive evolution, meaning that traits that enhance survival or reproduction tend to increase in frequency over time. So pretty much saying that natural selection results in evolution due to adaptation. The second mechanism we mentioned was genetic drift. We will spend more time discussing this mechanism of changing allele frequency since it's a new concept. Natural selection we have already talked about immensely in chapter 22. So that's why we'll now spend more time talking about genetic drift. What is genetic drift? They are chance events, keyword there, chance events that lead to fluctuation of allele frequency unpredictably from one generation to the next, especially in small population. This is contrary to natural selection. Natural selection can be predict depending on whether there is a trait that is particularly suitable for this environment. Whereas genetic drift, we're talking about chance alone. What do we mean by that? So first of all, this type of genetic drift may lead to a loss of an allele from the gene pool. It can be detrimental, completely wipe out of an allele, depending on how much of that chance have wiped out that particular allele. But which allele is lost, like I said, is a matter of chance. It's not like we are preferentially saying this allele is being favored in this environment. It's just random, okay? Is by chance whether it will be the one that survives, okay, or will be the one that prevails. So we're talking about events that associates with survival and reproduction are random, and it cannot be predicted, can never be predicted. So let's go back to the wildflower example we used earlier when we were discussing the Hardy-Weinberg equation. And let's say by chance, unpredictable, by chance, a large animal like a moose happened to stomp and walk over this wildflower population, and 3 of the CWCW individuals were killed in the process for generation 2. So this chance event, unpredictable, just this moose happened to stomp by, killed CWCW individuals in a small population, and then increased the chance that CR will be passed to the next generation because you killed the CW off. So now the CW allele frequency dropped even lower because it was 20% earlier, and now it's even lower because they have died. And then the CR population, which is the red, or CR allele frequency, which is the red pigment, would then have a higher chance of passing to the next generation. This means that these CR and CW change their allele frequency. So sometimes, by chance alone, only certain genotype flowers will give offspring. Okay, so in this particular scenario, we're talking about a catastrophe that has, by chance alone, have killed some of the genotype individuals off. Second reason, by chance alone, why we have genetic drift is that maybe certain genotype flowers will give offspring, certain will actually die, will not give fertile offspring. As remember, it's not just survival that we're talking about here, but also reproduction. So those that can give fertile offspring can lead to increased frequency in one of the alleles. Okay, so for example, this picture was given by the textbook. So let's say by chance in this first generation of the wildflower population, we talked about only 5 of the wildflower plants in the first generation were able to produce fertile offspring. So those highlighted in yellow, here, and here, these highlighted in yellow are the only ones that were able to produce fertile offspring. The rest don't produce offspring. So they're not going to have anything to do with the next generation. Then again, let's say in the second generation, by chance alone, only 2 plants of this second generation were able to produce fertile offspring. The rest of this, they don't produce any fertile offspring, okay? So then ultimately, by generation 3, this wildflower population has become 100% red flowers due to genetic drift, okay? It's not that red flowers gave it more preferential survival in this particular environment. It's just by chance alone that this were the 5 that produced fertile offspring, the rest didn't. And then these were the 2 in generation 2 that produced offspring to create generation 3, which is then all red, okay? Just by chance alone. I'm not saying red will be the one that is by chance alone be able to produce fertile offspring. It can be all the pink flowers can only produce fertile offspring, or white. And then depending which one can produce fertile offspring, that will lead to a change of allele frequency. In this case, actually wiping out your CW allele frequency by genetic drift, which is why we said drift may lead to a loss of an allele from the gene pool. In this wildflower population's case, the white allele or CW allele is completely lost, again by chance alone. So now, what general circumstances can result in genetic drift? We just talked about define what is genetic drift. It can be by chance events that affect survival, or it can be chance effects that affects reproduction. But what circumstances can result in those chance events? Turns out there are two circumstances. First, genetic drift can happen by what's called the founder effect, which is when few individuals become isolated from a larger population, and this smaller group may establish a new population whose gene pool now differs from the source population. In other words, this particular smaller group of the original population became the founder of the new population at another location. So that's why it's called the founder effect. And which alleles go with which smaller group of founders are completely by chance. So we can better see what we mean by founder effect in this particular example. So few members of the population, let's say this is your parent population right here, where you have a mixture of orange and gray color butterflies, okay? And a storm came through. A storm came through, blew a small group that had only orange butterflies off to a new location, isolated island. And then another small group got blown off, where you have a mixture of orange and gray butterflies that is kind of similar to the original population as we've seen here, okay? And then let's say another small group of butterflies were blown off, where you have the third small group being only gray color butterflies. So those orange butterflies, let's say, like I said, got blown off to an isolated island, and will only, if they were to start a new population from this particular subgroup, they can only produce orange butterfly offspring because they don't have any other alleles to choose from. Now, of course, we must consider that, let's say all of these are homozygous orange, or homozygous for orange color, because if this was, in the sense that if this was dominant recessive type of genetic pattern, because if this, let's say orange was dominant and somehow some of these are heterozygous, then you will be able to produce some gray butterflies maybe. But let's say it was not, okay? Let's say all of these orange butterflies are homozygous for this orange color coloration. So you can see how the small population that was separated by chance, this storm blowing the butterflies off, did not necessarily reflect the allele frequency of the original population. The original population looks kind of like 1-to-1, where you have 50% orange, 50% gray. But for this subset of orange butterflies, they will only produce orange butterflies. For this subset of the group that get isolated in another island, can only produce gray, and then this one will be the one that more so represents the original population. So it is completely random which small group would it be when we're talking about a storm that has happened to come by to swoosh away some butterflies. Hence, the change of allele frequency has occurred in these three founder population in the new locations, okay? So, or if we're talking about another example with humans, a British colonist was a carrier, let's say, for a progressive form of blindness called retina pigmentosa. And as a result of this colonist that was being a carrier, 4 of his 240 descendants had retinus pigmentosa. retinitis pigmentosa. So the allele frequency, in a sense, became 10 times higher in this particular colonized area. So the allele frequency for the retinitis pigmentosa was actually very low in the original population, in this case, Great Britain. But because the founder person, which is this British colonist, started a new population elsewhere, he or she was a carrier for this particular disorder. So it has increased the allele frequency of the disorder for their descendants, changing what was it before in the original population. So the second circumstance that can lead to genetic drift is what we call bottleneck effect. What is bottleneck effect? We're talking about a sudden change in the environment, let's say a fire or a flood, may drastically reduce the size of a population. And again, certain alleles may be overrepresented, underrepresented, or completely absent among those that survive by chance alone. So even if the surviving population recovers in size, that's a given enough amount of time, the small, tiny little population has grown back again, it may actually exhibit a low genetic variation for a long time to come. They may not ever recover, depending on the case. So here's an analogy for why it's called bottleneck effect. So here in this picture is the analogy. Let's say you have a bottle full of blue color, white color, and yellow color beads representing the gene pool in this particular bottle here. Shaking only a few beads through the neck of the bottle is analogous to a drastic reduction in the size of population. Let's say this was a big fire that have killed off all these individuals in the bubble, and only this small little population has survived. So in this picture's case, it is completely by chance alone that most of the beans that survive are blue, those that were shaken out from the bottle. And then they are overrepresented in the surviving population, where you can kind of see that there may be 5 blues and only 1 white bead and no yellow bead. It's not that blue or white beads were more prevalent in this original population, because if anything, I see about maybe half blue, half white, and a little yellow. Well, this just by chance, somehow, when the bottle was shaken, it was just blue and the 1 white that came out. So it could very well, by chance, on the contrary, those beads that were shaken out could be mostly white. It's all by chance. We don't know. We cannot predict like natural selection does, because which color beads get shaken out is completely random, just like bottleneck in this bottle situation. That's why it's called the bottleneck effect. So one example of bottleneck effect is the greater prairie chickens, which it was a population that actually decreased dramatically in size when prairies were actually converted to farmland, because humans will populate an area, they would then turn that area into a fertile land and produce for themselves in terms of agriculture. So when agriculture boomed in this era, you can it will actually wipe out the homes of these great prairie chicken. So from 1,000 to about 25,000 great prairie chickens, by the time it is post bottleneck time, you have only less than 50 chickens remained, because of majority of them losing their home and no longer able to survive. So in this case, this particular picture here you're seeing, because of human actions, habitat of these chickens that they best survive in are gone. Okay, that's why they're called greater prairie chickens, because they best survive in prairies. So hence their name, greater prairie chickens. And because prairies were converted to farmland, they have lost their habitat, most of them died, hence you can hardly see any green in this post-bottleneck situation. That's a drastic change. We're talking about almost, I would say, maybe 80% green to only 2 dots that are green. So the few that hatched and survive have very low genetic variation and also increased frequency of harmful alleles. So that's what, in a sense, lead to less than 50% of their eggs hatch, because genetic variation is a good thing, okay? Having genetic variation means you have more chance of alleles that can combat whatever environment it is in. But because it has now low genetic variation, that actually killed off a lot of their eggs. So the resolution was actually to bring in chickens from other locations to introduce more alleles into the population. 271 greater prairie chickens from neighboring states were actually added to this population in Illinois over the next 4 years after 1993. Thankfully, new alleles has entered into the population and the egg hatching rate has improved over 90% in just 4 years. Thankfully, it was able to be reverted by bringing in those other greater chickens with more genetic variations, and you're able to recover the population size. So, how bottleneck effect differs from founder effect is founder effect is not talking about a catastrophe that killed off its population. It's talking about a chance event that has moved the small amount of population to somewhere else. Whereas bottleneck effect, we're talking about a catastrophe that's killed off majority number of the population, and hence whatever surviving is the population you're starting with all over again. There's no migration whatsoever, where they're not migrating to a different area. They're not being blown off to another area like the butterflies we talked about earlier. That's how students a lot of times confuse between the founder effect versus the bottleneck effect. So here are the 4 key points about genetic drift. First, genetic drift is significant in small populations where allele frequency can be altered drastically, which is why when we talked about genetic drift by the founder effect, we have sort of got rid of, is now not a large population, but actually moved a small population into a new isolated area. That's why the allele frequency can be changed. Second, genetic drift can cause allele frequency to change at random. An allele frequency may increase 1 year or it may decrease the next. Don't know. There's no way to predict which one would actually be changed because it is all by random chance. Hence, the change from a year to year is not predictable when we're talking about genetic drift. Then that is contrary to natural selection. Natural selection is something we can predict because we know which traits will be favored in this particular environment. Or let's say even if they migrate to a new area, we know which traits will be unfavored due to the new environment. But genetic drift, we're talking about things are happening by chance alone. Third, genetic drift can lead to a loss of genetic variation within population. So we talked about this with the wildflower population. You started with a lot of genetic variation. By chance alone, those that can't produce fertile offspring will not go into the next generation. And then you end up with all red flower in the 3rd generation. So fluctuation of allele frequency can actually eliminate alleles from a population, which can lower the chances of adapting to the environment, kind of like the greater prairie chicken. In their case, they lost a lot of the genetic variation because now you have a very small size. They started with almost 80% covering Illinois state, and then they have decreased in size to dinky little red dots on the Illinois states. So that's really greatly decreasing in size of their population. In a small population, there cannot be a lot of genetic variation, which is why they lowered their chances of adapting to the new environment with all this farmland around, and they died. So fourth, genetic drift can cause harmful alleles to become fixed. And it can become fixed, meaning that they can reach 100% frequency and you can wipe out the species completely. So thankfully, this only happened in very small population, or at least a higher chance of happening only in very small population. And its survival can be threatened, kind of like the greater prairie chickens. There were only 50 left compared to thousands of them. Those 50 is a very small size. If they had not brought in that extra the greater prairie chickens from another state, That Greater Prairie chicken can be completely wiped out from Illinois because they did not have a very high survival rate compared with having greater genetic variation. All right, so here is a video that kind of, explain what is genetic drift and distinguish between and compare and contrast between the founder effect and the bottleneck effect, just in case you're not clear. I love Amoeba Sisters, by the way. If you want to watch more of their videos, feel free to search on YouTube.
So here's a question for you. Have you ever compared the candy you get on Halloween with your friends? My sister and I, a bit competitive in the sport of Halloween candy hunting, we both had preferences of which kind of candy we hoped to get in our buckets each year. And as for me, I love gummy bears. And every once in a while, someone would have them mixed in a big bucket, and I remember watching with intense focus the hand that picked out the candy, hoping for gummy bears. Getting those gummy bears was always total luck, total chance. Well, unless, of course, the entire bucket they had was actually gummy bears, but that would not be luck. No, that would be pure bliss. But that's never happened to me yet. So why bring this up? Well, what if instead of candy, we were talking about insects? And they weren't in a Halloween bucket because that'd be kind of gross. They're on the sidewalk. And when you're scooter riding through the neighborhood on Halloween to collect candy because scooters may or may not be our super awesome childhood mode of transportation, you squash some. Accidentally, of course, because you like insects overall. Were the ones that survived better adapted? Did they have some special ability to foresee events or to move your scooter away from them? Well, assuming they were all just there on the sidewalk in the path of your trick-or-treating, then no. In this random event example, the survivors were not better adapted. They were lucky. It was chance. And the ones that were squashed were, in this case, at the wrong place at the wrong time. And that's what genetic drift is. Genetic drift can change allele frequencies in a population. Remember, alleles are a form of a gene, but genetic drift is random, unlike natural selection. Recall that in our natural selection video, organisms that have traits that result in high fitness are able to pass those alleles on to their offspring. This is where that survival of the fittest comes into play, but not genetic drift. With genetic drift, the organisms aren't necessarily more fit. They just have won the game of chance. So let's look at a representation of genetic drift known as the bottleneck effect. I always like to remember this one by focusing on the word bottle. Well, let's say that we have a bottle here and we fill it with our Halloween candy. If we shake it out and we only get a few of the candy pieces, does it necessarily represent the whole bottle as far as the candy frequencies you got out? Nope.
No.
In fact, one of the types of candy pieces isn't even there. That type is totally eliminated. We'll consider a natural disaster, such as a forest fire. The surviving organisms weren't better adapted. They were likely just in an area where they weren't directly affected. But the survivors don't happen to represent the original population, so there's definitely going to be a new allele frequency among the surviving population. Bottleneck effect. Another time to see genetic drift is when you're talking about the founder effect. In this case, you may have organisms that have founded an island or some new area. The few organisms that arrive to start a new population do not necessarily represent the original population that they came from. Let's take a case where maybe some seeds that are dispersed by wind end up in a new area that happened to be one that was ideal for their growth. This new population grows. Well, those seeds that landed in this new area may not necessarily represent the original population of plants from which they came from. It's a random sample, founder effect. In summary, genetic drift can make some big changes to allele frequencies, and it's a mechanism for evolution. So one last thing. Which type of population do you think would be more vulnerable to genetic drift? A big population or a small population? If someone is just doing some intense candy hunting and by accident scooter rides over a few insects, look at the results in the small population versus the big population. As you can see, this random event has the potential to cause more change from the original in the small population. See, there weren't as many representatives there in the first place. So in many incidences of genetic drift in populations, the impact on small populations is especially significant. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so we are now again at 1 hour and 4 minutes. Again, I already said in the beginning of this chapter that this is a longer chapter. So we don't always have more than 2 hours of lecture, but this one is just longer and not that much longer. So I'll come back to you in part 3.